Morning, gang. So today, thought I would record this lesson, and then I will be available online during class time if you have any questions. All right. So we have 2 weeks left before Thanksgiving, and so that means 2 weeks left in this block before we are live in person. So the goal this week is to get through enzymes, into a little bit about how water moves in the cells, and then that's really it for biochemistry. Okay, and then we will be set up for when we are in person to get right into it and hit it pretty hard with photosynthesis and making ATP in the cell. Okay, cellular respiration. Had a brain fart there. Sorry about that. Okay, so let's get right into it. Okay, let's talk about enzymes. Okay, and so we're going to talk about what enzymes do in your body and how enzymes work and how enzymes are specific. Okay, now this picture I have there is just a drawing. Remember, enzymes are proteins. Okay, and then this enzyme you can see would be, remember we talked about the levels of protein structure, that would be a quaternary protein, right? The yellow and blue represent two different proteins that have come together, right? So remember, you have more than one protein coming together to make a functional unit, okay? So that picture, a drawing, okay, but you can see in there, you can see the coils, you can see there's folding, right? That isn't just like a bowl of cooked spaghetti, okay? That has a specific shape. All right, and that is important, and you'll see that as we go because enzymes are specific. All right, we have something called enzyme substrate specificity. Okay, certain enzymes only fit certain substrates, right? You can think of it, and now if you watch some videos and stuff, they've gotten away from this a little bit, but the easiest way to imagine this is like a lock and key. Right? You have a ring full of keys. Like I've got a whole bunch of keys on my key ring, right? You know, this key opens the male bathroom door, not the female bathroom door. Okay. You know, you wouldn't want to, you know, lock and key specificity, right? That's the bathroom I have to use. So that's what I have, right? Or this one unlocks the science office. They don't give me a key for the English office. We don't want to, you know, mix those subjects together too much, even though I try sometimes. All right. So important to know, we have specificity when it comes to that sort of thing. Apparently, I'm choking at the same time. Excuse me. So what are enzymes? Enzymes are protein catalysts that speed up chemical reactions by lowering the activation energy. Okay? Enzymes are not used up during a reaction. So enzymes can be recycled over and over and over again. Okay? So enzymes, they catalyze reactions. Okay? And then once, well, you'll see it's easier when you see it in a picture later, okay? But you know what, I gotta jump ahead for a second. What you get is this, right? So your enzyme, okay? So here is a chemical reaction. So your enzyme basically grabs onto your substrate. Sometimes it's two things coming together, sometimes it's one thing coming apart, okay? And basically they lower the activation energy, it spits out the products, they're recycled over and over and over again, okay? So you have, you know, those enzymes can get reused over and over. They're not used up, okay? Now that's not to say enzymes work all the time. They don't. You'll see that a little later, all right? Sometimes enzymes can be turned on to work more. Sometimes they're turned off to obviously not work, okay? Because sometimes you need your enzymes working really hard. Sometimes you don't need them working at all. Depends on what's happening in the cell at that time, okay? So key thing here, excuse me, they lower the activation energy of a reaction. So this is a good graph, okay? Something that you should understand. Okay, you see here progress of the reaction, free energy. Okay, so you see your reactants and your products, whether you have an enzyme or not, same amount of energy. What's different here, though, is the amount of energy that's required for that reaction to happen. Okay, enzymes speed up the reaction generally. Okay, or I should say they allow the reaction to happen with lower energy inputs, which is important. Okay, so you can see with an enzyme, EA is your activation energy, right? So with an enzyme, your activation energy is a lot less than without an enzyme. Okay, most reactions that are require an input of energy to happen, right? What happens is if you don't have that amount of energy, usually in the form of heat, right? That's the most common source of energy. You can basically have less heat to have your reaction happen because the enzymes speed up that reaction. Okay, so the enzyme here would grab your reactant or reactants, depending on what's going on. All right, lowers the amount of energy required for that reaction to occur. Once your products are released, that enzyme is now available to be used again. All right, and you'll see that some examples of that when we talk about DNA replication and protein synthesis and that sort of thing. You'll see these enzymes used over and over and over again. Okay, or if you're thinking about digestion in your body, okay, you have enzymes that get used over and over and over and over again. Okay, and those enzymes are always working, right? So as long as you're eating, those enzymes are working. And then enzymes are, I mean, you're full of enzymes, right? Enzymes make, they make you, right? You're basically a giant bag of proteins. And a lot of those proteins are enzymes that allow for, literally everything in your body to happen. Okay. So they're really, really, really important. There are so many examples. We're going to go over a few examples, right? But we certainly will not cover every example of enzymes in your body because there's just too many of them. Okay, so your enzyme substrate complex. Okay, now this is a three-dimensional kind of graphic. Well, it's not three-dimensional, sorry, it's two-dimensional. It is three-dimensional. I stole this picture off from online, and you can actually spin it to see that it has a three-dimensional shape. I should have kept that, but I have a little video after me that shows you this process, okay, that I want you to watch. So here is your enzyme, and it has a specific site called the active site. Okay, and the active site is where it binds to the substrate. Okay, and when the substrate fits in there, it's like your lock and key. They're specific. Okay, it goes in. Once it goes in, it causes a slight change in the shape of the enzyme. Okay, and that slight change just helps the substrate lock in, and then it can lower the energy required to, like I said, either break your substrate apart or put two things together, whatever the reaction might be. Okay, most enzymes are involved in, usually breaking things down. Okay, if we think of our body, digestion especially, right? Like you're eating things and your body has to break them down, right? But like we said, there are enzymes that are responsible for rearranging chemical bonds and two things in, two things out, right? But when we think of enzymes, a lot of time we think of things that break things apart, right? So it takes in whatever that one thing is, right? The induced fit, it just, when you get the lock and key that fit right, changes the shape a little bit, and that kind of puts pressure on the bonds that are in your substrate, and that's what helps break them apart with less energy, okay? So the enzyme itself is doing some of that work. Okay, so substrate, again, I'll explain it best I can, all right? Enzyme, substrate, maybe I'll be straight on here. Enzyme, substrate, it comes in, changes shape a little bit, and that little bit of shape change puts stress on the bonds that are in there, right? And then that's why it requires less energy, because the enzyme is stressing those bonds, so less energy is required to break them apart or rearrange them, okay? And that's where we have our enzyme substrate complex, right? So active site, your enzyme, your substrate. You can see here when they're together, see how there's that opening, it kind of closes on it. When it closes on it, it fits in right, closes, stresses those bonds, then they come apart with less energy, okay? And then it releases your products. That enzyme can now be recycled, okay? Here's that picture I showed you a second ago. All right. So in this example, you have two substrates. So this is catalyzing if you remember back from grade 10, right? Like like a double displacement reaction, right? It's not that simple in your body, but same sort of idea, right? You're starting with two things. Oops. You're starting with two things and ending up with two things, right? So substrates enter the active site, enzyme changes shape, so its active site embraces the substrate, induced fit. Substrate held in active site by weak interactions such as hydrogen bonds and ionic bonds. The active site, okay, and the R groups of its amino acids can lower the activation energy and speed up the reaction by acting as a template for the substrate orientation, stressing the substrates and stabilizing the transition, providing a favorable microenvironment, or participating directly in the catalytic reaction. Do I want you to memorize all that? No. Okay. What I want you to understand is when that enzyme grabs those substrates, right, it makes a good environment or it stresses the bonds. It just decreases the energy required for the reaction to happen. Okay. Excuse me, the substrates converted into the products. You release the products. Now that enzyme is available to react again. Okay. So pretty straightforward, not too crazy. Okay. So let's move on to some of the things that maybe we'll move on. Some of the things that affect enzyme activity. Okay. So, you know what? Slideshow from current slide. Let's do this. Okay. So, some of the things that affect enzyme activity, temperature is a big one. Okay. Temperature is a big one. So, all enzymes have an optimal temperature. Okay. Some are warmer, some are colder. So, you can see this enzyme. Okay. This is just, this is just a graph I got from online, okay? The enzyme is still working at 10 degrees and 20 degrees, right? But its optimal temperature is about 40, okay? After that, it peters out, okay? So enzymes have an optimal temperature, okay? Now, most enzymes in your body have an optimal temperature of, pretty close to this one here, right? Your body temperature is about 37, roughly. Okay, so this graph is pretty representative of the enzymes in your body, right? Obviously, if your body temperature is way down here, you're in trouble. You're probably going to be dead of hypothermia. If your body temperature is way over here, again, you're probably going to be dead. There's not much need for your enzymes to work well at really low or really high temperatures, right? And by the way, every single protein doesn't work well at the high temperatures, right? They become denatured. Think of an egg, right? If only someone gave you a question like that already, right? Your egg white, when you cook it, becomes, well, it goes from clear to white, right? Your egg whites, they're clear when your egg comes out of the fridge. They're white when you cook them, right? They become denatured. So that specific three-dimensional shape unwinds. And when it unwinds, it goes white. And obviously the properties change. It's no longer liquid, right? Your egg white is solid because it's become denatured. Same thing happens for an enzyme. But when an enzyme becomes denatured, they just don't work anymore, right? So if you're talking about, enzymes that are involved in ATP synthesis in your body, right? That would be bad if they became denatured, because what then happens is, what then happens is well, you're not making ATP, you're going to die. Okay, so here we go. Here's the explanation why. And I hate reading slides, but I'm going to read this a little bit, okay? So increasing the temperature increases the kinetic energy that the molecules possess. Okay, so in other words, when you warm it up, the chances of your substrate and your enzyme running into each other increase because they're moving more. Okay, so that's why your enzyme activity as you go up increases. The likelihood of them bumping into each other increase. And if your substrate and your enzyme bump into each other, boom, your reaction happens. When it's colder, they're not moving as fast, so they don't bump into each other as much. But, okay, if you get too warm, all right, what happens is those enzymes, they're complex three-dimensional structures. Remember, we mentioned they're quaternary proteins, right? You have lots of hydrogen bonding. You have lots of these things called disulfide bridges. There's some ionic bonding happening in there. So what happens is as your temperature goes up, those bonds basically fall apart, okay? Because the molecules themselves, the enzyme itself, as it warms up, all those atoms in there start vibrating more because it's warmer, 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 warmer. And as those molecules vibrate, right, those bonds break. And when those weak hydrogen bonds and the disulfide bridges and the weak ionic bonds, as they break, your active site loses its shape. Okay, so again, think of a lock and key. A little bit of an outdated reference, but this helps, right? Because if you think of your lock changing its shape a little bit, now the key doesn't fit in right. It's basically what happens. Okay, again, think of an egg, right? You put it in the frying pan, you wait a minute, 2 minutes, 3 minutes, it goes from clear to white. The proteins have fallen apart and they're now solid and white. Color change, chemical reaction. You remember that from like grade 9, right? Same thing happens here, right? Your enzyme falls apart. It starts to unwind. And when it unwinds, you lose that active site. And if you don't have the active site, you have nothing to catalyze the reaction. So your enzymes become inactive, okay? So another one, pH, okay? This is just another temperature, just to show you some enzymes have different optimal temperatures. Depends what it's in, right? That's the only reason I put that graph in. But I want to talk about pH next. Okay, there are optimum pHs, and those pHs change depending on what enzyme we're talking about. Not every enzyme is the same, okay? Now, most of the enzymes in your body, okay, fall into this category here. So this is amylase. So basically, the enzymes in your spit, saliva, okay? So those enzymes work best at about a pH of 7, slightly above 7. And that should make sense. Like you are just slightly basic, okay? Don't be offended by that. You're slightly alkaline. You have a pH of slightly above 7, okay? You're not basic, okay? I'm not basic. We're slightly alkaline. So most of the enzymes in our body work optimally at a pH of around 7, okay? Your stomach, right, is pretty, your stomach is pretty acidic, right? Stomach acid. So pepsin, which is an enzyme that is in your stomach, it works optimally at a really acidic pH, right below 4. And again, that makes sense because your stomach is a acidic environment, right? So it would make no sense to have the optimal pH of pepsin being up here with amylase because the conditions in your stomach are acidic, okay? But you can see, just like temperature, same sort of thing, okay? pH too low, the enzyme doesn't work, okay? It gets denatured, okay? Remember, acids can denature a protein. If you've ever, you can actually, if there's a Spanish seafood dish called ceviche, right? Where they take raw seafood and they cover it in like lemon and lime juice, all right? And when you do that, you cook the seafood, right? So if you look at a shrimp that's been in lemon or lime juice, it looks cooked. It looks white. It's no longer that kind of uncooked shrimp's not clear, but it's kind of that really, it doesn't have a lot of color to it. Versus cooked shrimp is almost white. That's what happens if you put a shrimp in lemon and lime juice. It's delicious too. It essentially cooks it, right? It denatures the proteins in it because it's a really acidic environment, okay? And the same thing will happen if you expose it to a higher pH, right? Remember, bases are just as dangerous as acids, right? You wouldn't want to mess with drain cleaner. It would hurt you, okay? So the same sort of thing happens with pH. There's an optimal pH. pH is too low, doesn't work. pH is too high, doesn't work. Goldilocks, just right. Your pH, your enzyme works the best, okay? But like we said, unlike temperature, most enzymes in your body basically all have the same optimal temperature because we're warm-blooded, right? pH, different. Most of them are about the same, around neutral is ideal, right? Except for the ones in your stomach, okay? It doesn't make sense, like we said. They are optimal at a low pH, okay? This year, let's say there's fish that live in the Antarctic, okay, or in the Arctic Ocean. Your enzymes would, well, you're not a fish. The enzymes in a fish that lives in the Arctic Ocean where the water is below zero would have an optimal temperature much lower than the enzymes that are in our body, right? Because those fish have adapted to live in 0-degree water. Okay, so that would be an example of where you would have enzymes that would have a lower optimal temperature. pH, like we said, most are around neutral. Pepsin is the exception there, right? Okay, and that's just what I said there. And for the same reason, right? You change the pH, it denatures the protein, so you don't get that fit anymore, right? You change the active site. And again, that's because lots of hydrogen bonding, right? And so when you start messing with acids and bases, remember, H plus for bases, OH minus, right? Hydroxides, those free H's and those free OH's kind of are floating around and start messing with the hydrogen bonds. And it breaks those hydrogen bonds, right? Because hydrogen bonds aren't especially super strong, right? So that's the reason why, okay? Substrate concentration, okay? So just simply, the more substrate you have, the faster the rate of reaction, okay? But you're going to reach a plateau, okay? So in other words, at some point, you're going to run out of enzymes, okay? Because remember, you don't have an unlimited number of enzymes. Your enzymes get recycled. So you increase the amount of your substrate. It goes up pretty quick until you basically are now occupying every single enzyme that you have, okay? So as quickly as the enzymes catalyze a reaction, they grab onto another substrate because they're flooded with substrate, okay? But this thing it doesn't increase exponentially forever, right? It peters out when your enzymes have all been used up, okay? And the same thing for enzyme concentration, okay? You increase the amount of enzyme, right? Again, you're going to reach a point where you've run out of substrate. So you only have so much substrate, okay? So same type of result. Goes up relatively quickly until you hit that kind of point where there's just no more substrate, right? Like you have a limited, if you limit one and have unlimited of the other, you reach a point where you just hit a steady state. And that's what happens here, okay? So, yeah, all right. You know what? I think that's where we're going to end for today, okay? No, we're not. We're going to keep going here. So cofactors and coenzymes. So some enzymes require a little bit of help. Okay. So coenzymes and cofactors. Yeah, you know what? No, we're going to end here. Okay. We'll get into this tomorrow. All right. I've gone on for about 25 minutes for a 40-minute class, and I want you to watch another little video. So we're going to leave this for tomorrow. So here's what I want you to take out of this lesson. Okay. You should know, okay, about enzyme substrate specificity. So specific enzymes work for specific substrates. You don't just have a body full of enzymes that work for everything. They're specific, okay? You should understand induced fit, right? Once the specific substrate bumps into the specific enzyme, okay, the substrate changes the shape a little bit. And that slight change of shape, that's what allows the activation energy to be decreased. Okay, most times it's because it puts stress on the bonds that are in your substrate. And so it requires less energy to break them apart, okay? Or less energy to form new ones because you've changed the shape a little bit. You've stressed the bonds, okay? And remember, energy is required to break bonds. Chemical bonds have energy. So to form new bonds or to break old bonds, either way, you require energy. That's what your enzymes do. That's what your enzymes are reducing, the amount of energy that's required to make new bonds or to pull those bonds apart. All right. And the enzyme stresses those. Okay. Lastly, you should be able to tell me intelligently the four factors that affect enzyme activity. Okay. Temperature. There's an optimal temperature. Okay. Remember, for us, it's about body temperature. If you're a fish in Antarctica, it's going to be a lot lower because you're never going to be warm. Okay. pH, optimal pH, right? And again, for us, most enzymes are about neutral, maybe slightly basic. Okay. Example of one that would be much lower would be pepsin in your stomach. All right. Substrate concentration and enzyme concentration. And those two increase until you plateau. Okay. That's what you should be able to tell me after today. All right. You should intelligently be able to tell me that stuff. I'm going to end it there. I have one other little video that I want you to watch after me, and then we will finish this up tomorrow. So I only have a 40-minute lesson. All right. That's it for right now. If you have any questions, I will be online during class time, and you can always email me. Bye for now.